Okay, today we're doing the cow eye dissection, and let's begin by looking at some of the external anatomy of the cow eye. We're going to start with the outermost structure right here, the most anterior structure, and this is the cornea. In an actual cow, this would be extremely clear like glass, but since this eye has been preserved, the preservative has denatured the proteins and caused it to turn cloudy. Around that, we see a lot of periorbital fat and connective tissue. And this connective tissue and fat plays an important role because this eye is located within an orbit, a bony orbit. And if you turn your head quick or you got a blow to the head and you didn't have this periorbital fat and you did not have all this strong connective tissue, then the delicate structures of the eye would easily be damaged. Let's look at the posterior surface of the eye, and we see this long tube-like structure coming out the back of the eye. That is the optic nerve, and that will go straight to the brain. Some of the other structures that I want you to see here is just some of this brown material here. You see a little bit of brown material over here, and you can see pieces of it here where it's been cut, and also over here a little bit. Those are eye muscles. Those allow the eye to actually move up, to move down, to move left, to move right. Now let's go ahead and begin by removing some of this periorbital fat. So I'm just going to take the scissors and I'm going to attempt to cut some of this fat and material off, sort of clean up the eye a little bit so that we can see things a little bit better. It's starting to look a little bit better. And that looks good there. We'll move some of this material out of the way. And you can see the optic nerve much better now, coming out the back of the eye. We'll talk a little bit more about that in just a few minutes. We want to start the internal dissection by cutting the eye right just posterior to the cornea. Now under some of this connective tissue is going to be another layer called the sclera. And we'll see that a little bit more clearly after we cut into the eye. So what I'm going to do here is I'm going to take a scalpel and I'm going to hold it a certain way because I don't want the scalpel to slip. And this sclera that I was talking about, this material, is very tough to cut through. So I'm going to take the scalpel and I'm going to hold it in the palm of my hand like this so that the handle cannot move backwards. And I'm going to put my hands around it, just like that. And I'm going to take the scalpel. Be careful to have your fingers on either side of the eye. And I'm going to put it just posterior to the cornea. Maybe right around there. And I'm going to gently push straight down. You can see that the eye is, there's liquid inside the eye. And this liquid and jelly substance inside the eye helps maintain the shape of the cornea and maintain the inner shape of the eye. We'll take a look at that. When I cut through here, you're going to see some liquid actually come out. And that liquid is called aqueous humor. And that liquid, one of the things that liquid does is it helps maintain the shape of the eye. We certainly don't want wrinkles in the eye as light is passing through it. So let's go ahead. I'm going to push down gently, straight down, straight down. There we go. And you can see some liquid come out. You can see the liquid begin to come out. And all I'm going to do is roll the eye and gently cut. Gently cut while I'm rolling the eye. Be very careful of your fingers here. If you want, you can use a pair of scissors to also do this. I find it a little bit easier to use a scalpel. And And be sure not to disturb the inner structures of the eye when you're doing this. And I'm going to open that up. I'm actually going to cut this right into two pieces. And let's take a look at the posterior aspect of the eye first, and then we'll look at the anterior part second. So I'm going to move this out of the way. And all the liquid that you're seeing right here, this is aqueous humor. The jelly-like material, that is vitreous humor, and it serves the same function. It serves to help maintain the shape of the eye. So we can see that there. What I'm going to do is I'm going to gently try to get rid of some of this vitreous humor, just kind of pour it out, nudge it out the side of the eye. And then I'm going to take a paper towel and I'm just going to dry this up just a little bit so that we can see clearly in here. And we can see this cream-colored material that coats the inside of the eye. This cream-colored material is the retina. This material is probably the most important part of the eye as it contains all the photoreceptors, the light detectors that actually detect the incoming light. Now as I move this material, as I move the retina, you can see how I'm bunching it up and I'm moving it. It's actually going to be stuck to the eye at a certain point, right here. We'll see this in just a minute. But one of the things I want to talk about here is what keeps this retina pressed against the inside of the eye. The vitreous humor actually does. The vitreous humor is going to press that retina against the inside of the eye. And when the vitreous humor is not present anymore, I can easily take this retina and I can just remove it and I can bunch it up. And you're going to see that when I bunch it up, it comes off rather easily, except in one place. In one place. And we're looking, there it is. You can see that one place right there where it appears as though the retina is passing through a hole in the eye. What that is that's actually the axons of the nerves in the retina. All the axons of the nerves in the retina are passing through the back of the eye. And where they pass through right here, the spot in the retina where they actually pass through is referred to as the optic disc or blind spot because light cannot actually be detected there. Now all these axons pass through the back, and if we look exactly opposite of that optic disc, that is where the optic nerve, all those axons are going to make up the optic nerve. That is where they exit and go to the brain. Now going back to the eye again, that's the retina. The next part that we come upon is going to be this iridescent, metallic looking structure here. Now humans don't have this, but animals that are nocturnal, that are active at night, or animals that are corpuscular, active during twilight, early morning, late evening, that actually helps them see better at night. That actually reflects additional light and allows that additional light a second chance to strike the retina. Humans, however, do not have that. This structure is called the tapetum, Now if I remove that, and actually the tapetum is going to be continuous with this other black layer you see here, that's called the choroid. The choroid is very vascular, contains a blood supply that serves the eye. The black color of this choroid also serves to absorb light. So when light comes into the eye, a lot of it is going to strike the retina and get absorbed, but extra light that passes between the nerves of the retina is actually going to strike this choroid and get absorbed, so it's not reflected around the inside of the eye and we don't have blurry vision. Under the choroid is going to be a thick white layer. Now it's stained a little bit black from the choroid, but I'm going to get the forceps in here and you can see how thick it is. I'm going to peel back a little bit of the connective tissue here and you can see this very thick layer. That thick layer is called the sclera. And you can see as I push in on it, it actually springs back to its original shape. Very, very tough material, and that's why we had to hold the scalpel a certain way when we began to cut the eye. Now let's move on to the anterior part of the eye. And again, I want to remove some of that excess vitreous humor. So I'm going to remove some of that very gently. Don't want to disturb the other structures of the eye. So very gently I'm going to remove some of that vitreous humor. And I'm going to take the paper towel and just very gently dab it a little bit so we can see clearly in there. You can see all that jelly-like material. Now, the first structure we notice is this gumball-shaped structure. This is actually the lens of the eye. This is the lens. It's responsible for, it actually changes shape, and it's responsible for focusing the light on the proper place in the back of the eye. Now, again, in a live cow, or you, this lens would be very clear, like glass. Now, this one's hard, but in a living organism, this lens would be very soft. You could actually squeeze it like you could a contact. Again, the preservative has denatured the proteins, made them cloudy, and made them very firm and rigid. Now let's take a look at this black structure that surrounds the lens. So you can see all these striations. That is called the ciliary body. And it is actually attached to the lens, and that ciliary body is going to be responsible for changing the shape of the lens. If we look close or if we look far away, that ciliary body is going to contract accordingly and change the shape, either flatten the lens out or make it more spherical like it is right now. I'm going to slowly remove the lens, and as I do, we're going to see if we can see where the ciliary muscle is actually attached to the lens. And you can see look, almost looks like saran wrap attached to the lens right there. You can see the attachment there. Those are suspensory ligaments, and they attach to the ciliary body, and that controls the shape of the lens. I'm going to slowly pull that out, and you can see those suspensory ligaments tearing in two. And I'm going to remove the lens. So I'm going to remove the lens. If we were to cut the lens, it would look essentially the same. Might look layered inside, a little bit like an onion. So I'm going to move that out of the way. And we're going to take a look at some of these additional structures in here. Just going to move some of that out of the way. And medial to the striated substance was the ciliary body, is going to be a more smoother looking black structure. That smoother looking black structure, and I'm going to take the forceps and I'm going to grab it, is called the iris. That is responsible for allowing more or less light to enter the eye. During the day you want less light, and at night you want more light. So that iris is actually going to change shape, and it's going to allow this hole here called the pupil to become larger or smaller. That hole in the iris is referred to as the pupil. In cows it tends to be oval, and in humans it tends to be round. And then if I look through that, I see the cornea. That was the structure that we looked at initially right here. That's the cornea. And those are going to be the structures of the eye. Now I want to go over one more thing, and that was if I take the lens and I replace the lens back in the eye, anything anterior to the lens, meaning where my probe is located, is referred to as the anterior cavity. Anything posterior, where my probe is located now, is called the posterior cavity. The vitreous humor is in the posterior cavity. And that's going to be it for the cow eye dissection.